interesting topic of gene profiling. Knowing the intricacies of our genes gives us a blueprint of our genetic makeup, giving us knowledge and understanding. Most importantly, allowing us to be proactive instead of reactive. So enough from me to you, Jules. Thank you so much.
Thank you so much, Jess and Stacey, for inviting me. And it's great, although you say 25 years in practice, I'm giving away my age then if you say 25 years in practice. So, but yeah, it's time. I think we've had a few lectures on the more functional side of health. And I just thought it was time for us to dive into the genetic side, seeing it really is quite a big part of functional medicine. But before I get into the talk, I just want to draw your attention to the disclaimer. So please note that the information on this webinar is for educational purposes only. So please, if you're going to make any changes in your health, please consult with your doctor. And this information is not designed to diagnose or treat any medical condition that you have. So I think genetics is, it's one of those subjects that seems so highbrow that very few people really know about it other than geneticists. So I'm going to, this lecture has got some parts that are quite technical, but I'm going to try and distill it into something that is fairly easy for everybody to see and draw into their own lives. Now, as humans, as compared to fruit flies, we have around between 20 and 24,000 gene sequences. In fact, we're not far off from fruit flies because I think fruit flies have got probably 20,000 different gene sequences. So basically what a gene sequence is, it is, as you said, Stacey, it's a blueprint. So it's a series of building blocks that the body uses to make proteins. And by proteins, I'm not just talking about, the sort of the muscle type protein. I'm also talking about all your enzymes, your hormones. All of these are actually proteins that the body makes. And the body makes them the correct protein that you need using our gene sequences as the menu almost, or as the blueprint, as you mentioned. But what's particularly interesting is that even though as humans, we've got around about 24,000 different gene sequences, we've got more than 100,000 proteins. So basically what that means is one gene sequence, which might be thousands of nucleotides long, doesn't only code for one protein. It actually might code for 10 proteins. And what makes the difference as to what the protein is how the protein is folded. And I mean, that is a science in itself, looking at how clever the body is in folding protein so it gets the protein that it wants. But really what this means is that our gene sequences are not exclusive. But what happens is that what makes me, and you, is the way our proteins are, at least is the way our genes are put together. So what happens is that you get in our gene sequences something called a SNP. So a SNP refers to a single nucleotide polymorphism. And really what that means is the beads, I'll call them beads, the fancy name is nucleotides, that make up a gene sequence for a particular protein. If one is wrong, it's called a single nucleotide polymorphism. And the impact in the body with these SNPs is that it affects the efficacy of essential enzymes and metabolic processes. And sometimes people think, okay, is it a mutation? It's not really a mutation. It's a variant. And that's why what sometimes happens is even within a family, you might have one member of the family has a manifestation of a particular non-communicable disease, whereas the other sisters and brothers might not. And that's got to do with the SNPs that we inherit. And we can inherit 1,000 different SNPs, so 1,000 different variations that make us unique. But what is also particularly worth knowing is that our SNPs can be influenced. So whether or not our SNP is expressed in our body can be influenced by the food that we eat. It can be influenced by our hormones, it's influenced by exercise, it's influenced by attitude, and of course, it's influenced by stress response. And what the influences do is that they can create a situation where we might have a particular gene SNP that might, in certain situations, predispose towards a health condition. But because we have a lifestyle that allows that gene SNP to be expressed or hidden, will make the difference as to whether or not that particular condition is manifest in us or whether it's not. So what we can do, and what is very empowering about knowing about your genes, is that we can, through our lifestyle, we can minimize negative influences and we can encourage positive influences. So when we look at gene profiling, what we're looking at with gene profiling is we're looking at the SNPs, the single nucleotide polymorphisms. And in South Africa, we're looking at between 80 and 120 gene SNPs. There are obviously more, but what's happened with the companies that are addressing gene profiling is they've looked at the SNPs that have the greatest impact in terms of our health. So they've taken the ones that are clinically most significant, and those are the ones that they read. And those are the ones that we would look at if we're going to profile. So when you're profiling, you look at the gene SNPs, and then you couple that with a comprehensive case history, and it allows one to create an informed plan of action that is going to minimize the impact of the deleterious gene SNPs and maximize the impact of those gene SNPs that are obviously going to lead us towards health. But I think the other misconception is that if you have a gene, if you've inherited a gene, it predisposes you towards a particular condition. But in actual fact, there are two terms that I want to discuss. The one is called penetrance. And basically what penetrance is, it refers to the number of people that have got this particular gene SNP who exhibit the manifestation of the genetic disorder. In other words, who show the sign of that SNP. So for example, it would be, the penetrance would be 100% if 100% of people who've got a particular gene SNP have that condition. But that almost never happens. In fact, it's so rare. Most of the time, the penetrance is less than 100%. It's an incomplete penetrance. And that means that you might find that depending on what's the classified penetrance, may be anything from almost nobody to 80% of the people might have that manifestation. But there's always a percentage of people who've got that gene SNP where there's no manifestation. And the million-dollar question is why? So the sort of things that influence penetrance, other genes might influence penetrance, and also age influences penetrance. So just because you've got the gene does not mean that you are definitely going to have that gene expressed and you're definitely going to get the particular condition or the disease. And we actually have an enormous amount of power when we address the lifestyle aspect of our genes, because it's the lifestyle aspects that will influence whether or not that gene penetrance is going to be closer towards the 80% mark, or whether or not it's going to be closer towards the, maybe the 10% mark. And I'm going to address the BRCA gene a little bit later, because that's one that worries people. Angelina Jolie had both breasts and a hysterectomy because we were told that she actually inherited the BRCA gene. But the fact that you inherit the BRCA gene does not have 100% complete penetrance. So there are a lot of factors, for example, like other genes, and as I said, lifestyle. And I also wonder whether Angelina Jolie just didn't want a nice new set of breasts, and that's why she had the other ones taken off and has a nice new set made. The other one is expressivity. And what this means is that in the people who have the gene, they might express in different degrees. So it might be no expression at all. In other words, they have the gene, but they don't have the condition, or they don't express the condition or manifest the condition, or they have the gene and the expression might be life-threatening. And again, this might be influenced by your other gene SNPs. it's influenced by your lifestyle. And of course, as I said, it might be completely asymptomatic, in which case, you know what, you're obviously not going to worry about it all. And for the most part, we've got many, many gene SNPs that do not impact on our health at all. So again, with profiling, what we do is we look at the gene SNPs that are going to or might influence on our lifestyle or on our disease risk. So in the past, what happened in the past, if we were looking at people's risk of developing what I call non-communicable diseases. So non-communicable diseases would be something like heart attacks or cancer or stroke or osteoporosis. These are all diseases that you don't catch from somebody else, compared with something like COVID, for example. COVID would be a communicable disease. You catch it. The non-communicable diseases are very influenced by our gene SNPs. But in the past, what would happen if, say, for example, your mother had breast cancer and you wanted to evaluate whether or not you were going to get breast cancer, what had to happen is that you could do nothing about it until you were already diagnosed with breast cancer. And then they would take a case history and they'd look at lab tests and they would treat the condition retroactively rather than proactively. And it was obviously a little bit of trial and error. Now, because we can profile our genes, we can treat our risks of non-communicable diseases proactively because our gene profiling is so personal and our approach to health is personalized now. So it's not to say, of course, I'm hoping that at some point everybody will be in a situation where they can profile their own genes at a reasonable cost. But in the old days, to profile your genes might have cost 20,000, whereas now the cost has come down and the cost is much more attainable for us if we want to see, for example, let's say, your dad has died of a heart attack or you've got breast cancer or cancer that runs in the family and you want to see what your risks are. Now you've got a better way of actually having a look to see, are you at risk? So when you have a look at SNPs in action, as I mentioned to you, there's small variations in the gene sequence and everybody has them. So you're not a strange anomaly or defective in some way if you have gene SNPs. We all have them, and how they manifest in our body depends on what the interaction is with the gene SNPs and of course with our lifestyle. And as I mentioned, it affects the efficacy of enzymes. And I just want to mention here that really enzymes are the workhorses in our body. Enzymes, we've got probably, in fact, I think it's about 1,000 different enzymes that act within our cells. And the enzymes are, without enzymes, we wouldn't last a second. We need our enzymes because they are what allows our metabolic processes to work properly. So gene SNPs, for example, play a big role in liver detoxification and methylation, and I will address those in a little bit more detail later on. And the effects are very far-reaching. So it's beneficial, if you can, to just have a look and see where you've come from in terms of your inheritance. But as I mentioned, just because you have a gene SNP, it doesn't mean you're definitely going to get that disease. So when we're looking at gene SNPs and we're profiling genes, we look at trends. So we look at propensities to develop a particular condition. So the sort of trends that we're looking at when we're studying gene SNPs, we're looking at your detoxification capacity. So for example, how well can you detoxify, let's say, pesticides or pollution? Let's say, for example, you inherit gene SNPs that don't detoxify well, and you smoke, and you live next to a highway, then your chance of developing problems in association with the detoxification capacity is going to be more than somebody who hasn't inherited those gene SNPs, who doesn't smoke and who doesn't live next to a highway. The other thing is oxidation capacity. So how well your body cleans up free radicals. And free radicals are very damaging to the tissue. So you want to be in a situation where your body is mopping up free radicals, and that is, your gene SNPs will also influence your oxidation capacity. It influences your risk of cancers, mood disorders. Mood disorders are very big because many people who have mood disorders, so let's say, for example, they've got depression, clinical depression, and they go on to SSRIs, so that would be selective serotonin reuptake inhibitors, and they find they don't work at all. And if you go a little bit deeper and you look at how the gene SNPs influence mood disorders, and you address the gene SNPs, then you have a better chance of the SSRIs working, or you may not even need the SSRIs. The other thing is inflammation capacity. And remember, all our chronic diseases have their grounding in inflammation. So cancer's grounding is in inflammation, heart disease, Alzheimer's, stroke, they all have a grounding in inflammation. So again, if you've got a lot of gene SNPs that indicate that you have poor inflammation capacity and you can address them, you are already mitigating your risks for the grounding of a lot of chronic diseases. Dietary influence, many people, diet is an issue and obesity is an issue. And nowadays, I think we're exposed to so much information about diet. Do you go on a keto diet? Do you bench? Do you calorie control? How do you go about losing weight? And what influence does your diet have? And we can have a look when we look at the gene SNPs, and we can see, for example, that this particular person, a keto diet is not going to be best for them because their body's not going to process the fats well. And then you would direct them in another direction. The last one to mention is cognitive risks, and that's the APOE4. And APOE4 is probably the primary gene SNP that influences your risks of developing Alzheimer's. So, and we do test this. So for those of you who are interested in having your genes profiled, it's one of the ones that we do test. And there are many, many ways that we can mitigate the risks of developing Alzheimer's. And in fact, Dr. Dale Bredesen is doing clinical trials on people nowadays who've already been diagnosed with Alzheimer's. They may be APOE positive or not, but he is turning it around where people who've been diagnosed with Alzheimer's, in actual fact, by addressing, I think it works out at about 36 different factors, by addressing those factors, and even if they're already diagnosed, he's turning their cognitive ability around. So it's very helpful to know this. In the gene profiles and the SNPs that we look at now, we look at issues like thyroid health, mood and stress that I've mentioned, Alzheimer's that I've mentioned, hormone detoxification. And this is important because with hormone detoxification, if you, for example, have a history of breast cancer running in your family, it might be because your body's not detoxifying estrogen. And you can have a look through your gene, and I'm going to address that specifically in this particular talk. If you look at your detoxification of estrogen, you can create a situation where your body's detoxifying estrogen well and reduce your risks of developing cancer. Insulin impact, how your body processes sugars, histamine sensitivity. So if you tend to be a snuffly person and you have a lot of allergies, we can see that from the genes. Intestinal permeability, so how intact your intestines are so that your bodies are not picking up the gut bugs that should stay in the gut or partially digested food, which would trigger an immune response. Vitamin D impact, and I'm sure that many people who are listening to this particular talk now, we're all aware that vitamin D is playing a big part in risks of catching COVID and risks of how the impact of COVID influences you. So vitamin D is important because certain people have a gene SNP with vitamin D that means that the receptor site is not very sensitive. So for them, they literally have to pummel their body with vitamin D to get the same effect as somebody who doesn't have the particular gene SNP. Cholesterol impact, and of course, I have mentioned inflammatory risk. So with knowledge and understanding, we personally have got the power to influence the expression of our genes. And that means that for us, looking forward, we're no longer at the mercy of particular non-communicable diseases where you think Alzheimer's is a big one, where you think, well, you know what, I can't do anything to help myself. Because in actual fact, there is something that we can do personally to change that whole profile. All right, so as far as genetic testing is concerned in South Africa, there are 3 companies in South Africa that do it. They're all very good companies. It costs normally between 4,500 and 5,000 Rand to get your genes profiled in South Africa. Now, although you do get a report from all of these companies, the report, which I'm going to show you now, is probably incomprehensible to the average person. So for the most part, your genes need interpretation. So once you've had your genes profiled, it does need somebody who understands the interaction between the gene SNPs to profile those genes for you, sit down with you and explain to you what is going to be the impact of certain genes for you, and what can you personally do to change that impact in your body. And an interpretation will cost anything between 750 Rand and 1,500. And really what this does include is that the practitioner needs to get your genes, look at your genes, go through your genes, and that normally takes approximately an hour and a half to 2 hours, then sit down with you and go through it. And that consultation is at least an hour. So it's quite labor and time intensive. All right, so this particular gene profile is from one of the companies called, my brain has just died. GeneWave, sorry. It's the GeneWell profile. So it looks like this. Now you'll see there are red dots and yellow dots and green dots. When you see a red dot on your profile, it means you've inherited that particular SNP, single nucleotide polymorphism, from both your mother and your father. And when you inherit it from both parents, obviously the impact on your body is going to be much greater than if you inherit only from one parent, where it's reflected as a yellow dot, or you've not inherited it at all, and then it's a green dot. So when it's from both parents, we call that homozygous. When it's from one parent, we call it heterozygous. And when you don't inherit it at all, it's called non-variant. So you can basically see what the impact is going to be, and then you can get direction on whether or not this is something that you can actually change. And they are all gene SNPs. All gene SNPs are manageable, and you can mitigate the effects. So for example, we look at things like gut health and digestion. We look at circulation. So in other words, what's going on with your heart health. We look at inflammation and immunity. And of course, immunity is very big at the moment. Structural and cellular integrity. So what's happening with your cell membranes? So for example, if you inherit a gene SNP called PEMT, basically what that does is it influences the integrity of the cell membrane. And if you think about it, we've got trillions of cells and of course trillions of cell membranes. And if you've got problems with the integrity of your cell membrane, it's a little bit like you've got a house and you want a nice structure so that the windows can fit nicely in the house and the door is fitting solidly so that, you know, somebody can't breach the house. But if your cell membranes are not, they don't have integrity, then your receptor sites don't work properly. So it's important for general health to make sure that your structure of your cells works properly. So just a few caveats. The whole subject of genes and gene profiling is really very multi-layered. There's so many variables that need to be taken into account that assigning causality, so let's say, for example, you do get breast cancer, assigning causality to a particular gene SNP is almost impossible because there's so many possible variables that may have triggered that manifestation of that particular condition. And it's impossible to say, okay, it was this or it was that. But what we want to do when we're looking at gene profiles and when we're looking at your health as a whole, so when we're looking, for example, at lifestyle, we want to marry what we see on your gene SNPs with what you're doing in your life. So as I said to you before, the example I gave with smoking, if you are a smoker, we all know people who get to 95, they've smoked a packet a day, or they've drunk a bottle of whiskey and they get to 95, and every person who smokes, who drinks, is convinced that they're going to be that person who's going to get to 95 and they're going to live and everything's going to be wonderful. But the reality is those people happen to have perhaps gene SNPs that allow their bodies to detoxify very easily. Whereas you might have gene SNPs that don't, and then you definitely are not going to be one of those lucky people getting to 95. So again, we're looking at the trends. We're marrying the nature, in other words, what you inherit, with the nurture, which is what your lifestyle feeds into your risks of non-communicable diseases. And the contributors are collective. So if, for example, we see gene SNPs and they are what I call, they're soft spots, they're areas that they put you at risk, and you decide you're going to address those. There's so many ways that you can address them that just allows you to relax a little bit and take the view that you are doing everything that you can to make sure that you're not going to get that particular condition. But of course, there are no guarantees, because I mean, if you think about, we've got 25,000 different genes, and we've probably got many thousands of gene SNPs. So, you know, although most of our gene SNPs do not have an impact on our body, finding out every single little impact and how every gene SNP impacts on another gene SNP at this point in science, they haven't got to the point where they can actually do it easily. So we do the best we can with what we know now to make sure that your risks are mitigated. So I could have picked Alzheimer's, I could have picked inflammation, I could have picked detoxification capacity, but I decided I would use cancer as my example so that you can see how the genes work, because, you know, I think we all know somebody who's been afflicted with cancer, if not ourselves. So I'm going to use cancer just to show how this is a multi-layered approach. So the contributors to whether or not we get cancer are genetics. Now, genetics actually only counts for about 4 to 5% of cancers. So that means that 95% of whether or not we get cancer is attributed to our lifestyle. So if we have a SNP mix, it might increase our susceptibility to cancer, but we can have a look at the lifestyle and see whether or not we can soften that blow. Obesity is a very big contributor to cancer. Smoking, alcohol, diet, environmental pollutants, and infectious diseases. Okay, so I'm not going to dwell on this. There will be a recording of this lecture, and then anybody can just go into it and have a little look at this. But if you look above the line, you can see alcohol. Alcohol contributes quite a bit to the risks of cancer. So when you look at liver, for example, for men, alcohol increases your risk of developing liver cancer by 18%. For women, it's 12%. If you look at smoking, for example, you can see the increase in risk is really, really very high. Esophageal cancer, for example, 54% increase in risk for men, 46% increase in risk for women. So it just shows how an external lifestyle factor can influence the risk. So obviously, you know, the obvious thing is don't drink and don't smoke. But if you are drinking and you are smoking and you look at your detoxification capacity, you may still get away with drinking and smoking if you've got a good detoxification capacity. And again, we'll look at that. As far as diet is concerned, overall, diet increases, obviously I'm talking about a poor diet, not a good diet, increases your risk of cancer by 35% overall. So when you look at diet and endometrial cancer, for example, a poor diet can influence your risk of endometrial cancer by 50%, breast cancer by 50%. So it's a lot. And that's why when people are ignorant about what impact a poor diet has, and they see the impact on cancer, for example, I think knowledge really does empower one. Because if you look at it in reverse and you look at the fact that a good diet might reduce your risk of developing cancer by 50%. Those figures don't actually work out. I'm not a mathematician, but they wouldn't work out quite that way. But so by changing one thing in our lifestyle, the impact on our body is huge. Obesity. So obesity for men increases risk of any of these cancers by collectively about 14%. For women, it's about 20%. So when you marry up the gene SNPs and the gene SNPs that lead one into a higher risk of becoming obese, you can see how getting to know your gene SNP might, or your gene SNPs might influence your risks of obesity. And if it influences your risk of obesity, the follow-on of this, it's also going to influence your risks of developing any one of these cancers. Infections. So if your immune system is working well, you're going to have a reduced risk of developing infections. So if infections play a role in getting cancer, which they do, and you've got a very effective immune system, that's going to impact on your risk of cancer. If you don't have effective immune system because you've got certain gene SNPs, then one needs to address that. You need to upgrade the impact of that. So what one wants to do is that you want to make sure that those particular gene SNPs don't express so that it does mitigate your risks. Environmental carcinogens. I mean, I'm sure we've all seen that movie, Erin Brockovich, if I pronounced that right. So environmental carcinogens, they are rife. And in fact, it's something along the lines of 80,000 different, we're exposed to something along the lines of 80,000 different environmental pollutants. And when a baby's born, they actually took umbilical blood and they tested it for environmental carcinogens, chemicals. And they found as the baby was born, there was something along the lines of 250 different chemicals in the umbilical blood. So we can't get away from environmental carcinogens. So what we have to do if we can't get away from it, and that includes EMFs, so electromagnetic fields, if we can't get away from it, then we need to make sure that our genes don't make it worse. Obviously, if we live in a situation where the water is polluted, or we live right next to a highway or any of those, and we are constantly bathed in environmental carcinogens, our chance of escaping it is probably not that great. But there are always things that we can do to try and soften that blow to our body. Okay, so at this point, I'm going to just pause for the moment before I go into the sort of how excess weight plays a role. Are there any questions that anybody can think of at this point? Nothing coming up now. Okay, good. All right. So I've mentioned obesity a couple of times. And if we look at the impact of excess weight, so weight and weight gain accounts for approximately 20% of all cancer cases. And being overweight almost doubles the risk of dying from breast cancer. So being in a situation where your weight is normal, or if you are overweight, finding a situation where you can lose weight, and I know that's easier said than done, it's going to impact very well on whether or not you have a risk of developing cancer. Being slim reduces the amount of circulating estrogen, and estrogen plays a role in whether we develop breast or endometrial cancer. And I'm sorry, I put this in, it's very sad, but there is no risk-free level of alcohol consumption. So not even half a glass of alcohol is an acceptable consumption. And the risk of breast cancer increases by 30% if we drink more than 3 glasses of wine, of alcohol really a day, which is terrible because, you know, when you get postmenopausal and your hormones are frazzled and you've got road rage and you're prickly and you don't sleep and you've got insomnia, all you want is that glass of wine. And I know you don't want me to hear me say that it increases your risk of cancer, but the reality is that it does. Insulin resistance. Insulin resistance is it has the single biggest impact on developing chronic diseases. So now I would go so far as to say, if you're going to make one change in your body, get rid of sugar and processed carbohydrates, because developing insulin resistance, and stress, by the way, affects insulin resistance, and genes and gene SNPs affect how we manage stress. So I'm hoping that what you can see here is that how the body's like a web, and all of these factors feed into our risks of non-communicable diseases. And working systematically through these risks, mitigating the risk, softening the blow, depending on what SNPs come up, they're all steps towards health that will allow us to live good, healthy lives until we're 95. We don't want to exist and just be alive until 95. We want to be healthy and be vibrant and be able to do all the things that we did when we were younger, and then hopefully by the time we get to 95 or 100, take 2 weeks and then, with 2 weeks and then die, rather than 20 years of chronic diseases where we're on pharmaceutical drugs and we just live a subpar life. That's not a way to live. So can gene profiling help? So gene profiling looks at which genes make you fatter, which genes can increase your risk of insulin resistance. So those are 2 things we can address that already, just from a cancer perspective, never mind anything else, just from a cancer perspective, if you address those, you're going to reduce your risk of developing cancer. Which genes affect your mood and your stress response? Because stress not only gives you belly fat, and stress makes you fat, but stress, if you have a situation where you're very anxious, stress affects the joy with which we can live our lives. So we want to look at a situation where if you are very stressed or you've got a lot of anxiety, what can you do to actually help yourself feel better? Which genes block or promote detoxification? Which genes block or promote immune support? Very, very relevant at the moment. Sorry about the spelling mistake. Do you have genes that clear pollutants, so like smoke or carbon monoxide? And issues like, do you need to address your thyroid to lose weight? But at this point, what I want to say here is that we can look at your genes, and because genes address the trend, we still need to see what is the manifestation in the body. So if you look at your genes when you're young, you can already see when you're young, okay, that's a soft spot. I need to make sure that as I get older, I don't put stress on that particular metabolic process in my body. But as we get older too, we need to see what is the clinical manifestation in the body. So I would recommend that about every 2 years, you go for baseline blood tests. Because, I mean, we service our car once or twice a year, but we don't service our body. And just to address this, we can catch all of these before they reach a point where the damage on our body is so great we can't turn it around. All right, detox in the liver. Let me just address detox in the liver. This is quite technical, but basically what happens is that when our body's detoxing a substrate. Now, by a substrate, I'm not only talking about a toxin, I'm also talking about hormones and what we eat and our vitamins and all that. They all pass through the liver. And there are 2 phases in the liver. So phase 1, the substrate. So let's take estrogen, for example. In phase 1 in the liver, estrogen is acted on by what we call a P450 enzyme. These enzymes are really, really important. And it's these enzymes that we measure when we look at our gene profiling. And it's these enzymes that may be effective or may not be effective. And if, for example, they're not effective because we have a gene SNP in this particular enzyme, it means then when our body is trying to detoxify, let's say estrogen, and it forms a metabolite, that metabolite might be more carcinogenic than the primary substrate. So for example, estrogen, when it's detoxified and it's acted on by a P450 enzyme, it forms a metabolite, and some of the metabolites are more carcinogenic than estrogen itself. So once the metabolite is formed, the body then shunts it to phase 2 in the liver, and then it's conjugated by another molecule. So once it's conjugated, then it allows the body to excrete it either in the urine or in the feces. And it needs various processes to join to that metabolite so that the body can excrete it. And phase 2 conjugation includes a process called methylation, one called acetylation, glucuronidation, sulfation, and glutathione. So methylation, I'm going to address the impact of methylation a little bit later, just so that you can understand the complexity of it. But all of these processes we address in gene profiling. So we address your methylation capacity when we're looking at gene profiling. We address your sulfation capacity, because if, for example, your methylation is substandard or your sulfation is substandard, your body can't detoxify estrogen properly. Estrogen then accumulates in the body, and the metabolites are more carcinogenic, and it collects in estrogen sensitive tissue, of which breast tissue and endometrial tissue are estrogen sensitive. And because estrogen is proliferative, it makes more cells, then you can develop tumors. So you can see if your detox capacity is good, your P450s, so your enzymes are working well, your conjugates are working well, your detoxification of pesticides, hormones, what else? Persistent organic pollutants, phthalates, these are all, which are like plastics. These are all environmental carcinogens that we're exposed to that we actually can't do anything about. We can't get away from them. So we want our liver to be able to detoxify them properly. And that's why, in actual fact, alcohol is a problem, because if you've got a high intake of alcohol and it affects the capacity of the liver to detoxify, then you can understand that if the liver's not detoxifying properly, you're not detoxifying your pollutants, they build up, and it increases your risk of non-communicable diseases. All right, just very quickly go through this. This is just phase 1 of estrogen. Estrogen actually goes through certain SNPs, so certain enzymes, CYP1A1, 1B1, and 3A4, and it forms metabolites. And when it goes through these enzymes, these metabolites are more carcinogenic than estrogen as it stands. And we look in the gene profiling. So for example, if you decide, yes, I want to do gene profiling, we're going to look at these enzymes, and we'll be able to see, yes, maybe, for example, this particular patient has got the CYP1B1 that forms 4-hydroxyestrogen, which is carcinogenic. And then the things that you can do to make sure that enzyme is upregulated, or in some cases, the enzyme are over upregulated and you need to downregulate them a little bit. So you need to see whether it's upregulated or downregulated, and you need to mitigate that effect. Similarly, in phase 2, methylation. So what happens with methylation? It's a carbon with 3 hydrogens attaches to the substrate, and it allows the body to detoxify. And we can have a look and we can see whether your methylation process is working well. And at this point, in fact, do I want to mention it now? No, I'm going to mention it just now. So let me just move on from that. But we look at all of these to see what your methylation process is like, or what your sulfation process is like. Oh, yes. All right. This is just a little joke, actually, because when I speak about methylation, this methylation map is not for you to know anything about or even take note of what it is, but it's just really, I put it in to show you how very complex the process of methylation is. So one does need to understand methylation, but I want to mention here, when it comes to risks of cancer, our genes, to protect them, are wound around, it's almost like a spindle. It's called a histone. And because our genes are wound around them, it protects our genes so they don't get damaged. And what keeps our genes pinned to the histone, so in other words, it keeps them pinned to the spindle, so they're not like those springs that just burst open and show all the genes so they can get damaged, are methyl groups. So methyl groups work like little pins to keep our genes protected. So for example, they keep our oncogenes from expressing, but we don't want too much methylation either, because we don't want our tumor suppressing genes to be held to not express. So methylation is a very complex process, and I would go so far as to say it's one of the most impactful variables that one needs to address when you're looking at genes. So let me stop at this point and see, are there any questions? Because I know some of this is a little bit technical. Any questions? I hope there are no questions, because I'm explaining nicely, not because I'm explaining so badly that nobody knows what's going on. All right, so let's drill down now to breast cancer and gene SNPs. I chose breast cancer. I could have chosen colon cancer or Alzheimer's or any of those, but I chose breast cancer because mostly the people who follow these webinars are mostly women, although I know that there are some men listening to this webinar. And women generally are more proactive about taking control of their health. So I decided, let me look at breast cancer, because I'm hoping that that's going to be informative for the people who are listening. So for breast cancer at the moment, the prognosis for breast cancer is exactly the same as it was 50 years ago. There's been zero advance with breast cancer. So in South Africa, approximately 1 in 29 women are going to develop breast cancer, which is very, very high, and that incidence is rising. So your risk increases with age. And if you're looking at other members of the family, so if other members of your family have got breast cancer, one always wants to ask the question, is it going to be me? So what were the factors that influenced my mom or my grandmother to develop breast cancer, and have I inherited them? And if you get breast cancer and you go to an oncologist, they do test for certain genes, but ironically, the genes that I'm going to talk about now that we look at, they actually don't test for. So these gene SNPs are gene SNPs that will help you prevent getting breast cancer, rather than when you've got breast cancer, treating the breast cancer. Obviously, prevention is much better than finding a way or working through your genes to actually treat them. Now, if you look at breast cancer and gene SNPs, these are all gene SNPs that are tested. But in the Journal of Gynecological Oncology, they did a study and they saw that the enzymes 1B1 COMT, the GSTPs, and the manganese superoxide dismutase, the combination of those increased the risk of postmenopausal breast cancer. So if you have one of those, it's not enough to kick you into a situation where you are at high risk for breast cancer. But if you have all of them, that's obviously going to increase your risk. And the good news is, even if you do a profile and you find horror upon horrors, you've actually got all of these. Every single one of those, there are lifestyle changes that you can do, you can adopt in your life that is going to mitigate the effects. And basically what it does is it prevents those genes from expressing, or when you think about expressibility, which I mentioned at the beginning, it keeps it at a very low expressivity, excuse me, that is the word, expressivity, it keeps it low rather than keeping it high. So the genes associated with breast cancer risk are all of these. I know that they're going to mean absolutely nothing to you, and that's why if you are having your genes done, you do need somebody to interpret them. But we do measure all of these, and all of these, you can do something about all of these. I do want to mention BRCA. Even though we don't test for BRCA, the reason we don't test for BRCA is actually because the impact on developing cancer is actually very small. So what BRCA1 and BRCA2 are, they're tumor suppressors. So if you have gene SNPs in BRCA1 and BRCA2, it basically means it stops the tumor suppressing effect. So you are more likely to express the tumors if you've got gene SNPs in BRCA1 and BRCA2. But only 3% of women with breast cancer have the BRCA gene. So that means 97% of breast cancer is caused by other factors. And that's one of the reasons why we don't test for BRCA really, because the 97% are all factors that we have more control over than whether or not we've got BRCA. But in actual fact, even if there was BRCA, there are things that you can do to keep BRCA to not express that. So the other factors that will influence whether or not you develop breast cancer: vitamin D, detoxification of estrogen, detoxification of environmental pollutants, obesity, and insulin resistance. And we really have addressed all of these. Vitamin D. Now, not only is vitamin D very good for our risks of the how severely we might be affected by COVID, but in actual fact, when they looked at studies, they found that low vitamin D, in other words, less than 20 nanograms, correlates with increase in breast tumor size. The overall survival rate improved as the vitamin D levels increased. In menopausal women, if they had low vitamin D, their relapse rate was higher, their metastasis rate was higher. So the higher the level of vitamin D you have, the greater the reduction in mortality and the better chance you have of surviving from breast cancer. And this is a good example where if we look at your gene profile and we see, okay, you've inherited that gene SNP that causes an ineffective receptor site for vitamin D. We now know that with you, we would then do a blood test to see what is your present status with vitamin D and see where you are with your present status. And then really for somebody who's got that gene SNP, we would really have to give them very high doses of vitamin D and check their vitamin D to make sure their vitamin D status is actually going up. So it's very measurable. But who would think? I can almost guarantee that probably very few people who are listening now routinely go and have their vitamin D levels checked. We think that because we live in South Africa and we've got this glorious sun, this sunlight, that our vitamin D levels are good. But what do? We go from our car into our office, although nowadays we don't even leave our house. We very seldom go out in the sun because we've been terrified by the thought that the sun exposure is going to increase our risk of skin cancer. But in actual fact, the opposite applies, because high vitamin D reduces the risk of all cancers, including skin cancer. So although melanomas are definitely something to be careful of, mostly melanomas come in areas that are not exposed to the sun anyway. But that's just a little aside. All right. I've mentioned we need to test and you need to supplement. All right, so clinically relevant SNPs, generally clinically relevant SNPs associated with cancer in general. These are associated with cancer in general. And again, as I said, I know that the name is not going to mean anything to you, but it just, I'm just showing you these so that you can see what we test. And I'll just go into now the impact that they have. So the GST that we test, that looks at detoxification. So these are enzymes that help the removal of all the xenobiotics, the carcinogens, oxidative stress products. So we want your GSTs to be working properly. The GST-Ms, this one is interesting because some people, they actually miss out entirely on these genes. So they have what we call a null genotype. They haven't got the gene at all, never mind an ineffective gene. They just don't have it at all. And these people have to be particularly careful to reduce their exposure to carcinogens because their body cannot detoxify them. And we do have a look at these so we can see whether or not a person has got these genes when we do the gene profile, whether they've got them but they're ineffective or whether they've got them and they are working properly. Manganese superoxide dismutase, this one addresses free radicals, and free radicals destroy our mitochondria. So our mitochondria are little organelles within our cells, and they put out our little energy pockets, so the ATP pockets. So without mitochondria, we would be dead within less than a second. But what happens now with cancer is that now they're looking at cancer as being a mitochondrial disease, not necessarily what we call a somatic disease. In other words, they're something that affects our DNA. They're now addressing mitochondria as a metabolic disease. So we want our mitochondria to be working perfectly. Sorry, it's cancer as a metabolic disease. So we want our mitochondria to be working perfectly. And if we've got a lot of free radicals, the free radicals destroy our mitochondria. And it's the manganese superoxide dismutase that helps to protect our mitochondria. So if we've got this gene SNP, we need to include in our diet the antioxidants. So antioxidants like vitamin E or vitamin C. We need to eat a diet that's got lots of colorful fruits and vegetables, that's very high in phytonutrients, because phytonutrients also help to mop up these free radicals. Salt, I mentioned salt. This one helps to detoxify estrogen. So when our salt enzymes are not working properly, it leads to the accumulation of harmful estrogens. So how our sulfation really, we can have a look and see what's going on with the sulfation. MTHFR, this is a biggie. It's probably one of the best tested gene SNPs of all of them. It's actually involved in folate synthesis, but it's a very good marker to see what the methylation process is in the body. Is your methylation working well? And we use this gene as almost like a surrogate marker to see what's happening with the methylation, because it's got such a wide health impact. So if you have a look at this list and you see, for example, MTHFR it plays a role in addictive tendencies, in anxiety and depression, chronic fatigue, our detoxification. It affects whether or not we can process histamine. It affects whether or not we're going to sleep properly. Excess stomach acid, so for example, reflux, our sensitivity to pain, whether we've got joint pains or muscle pains, phobias or paranoias, it influences those, whether or not you're OCD, obesity. So you can see, having a look to see we've got MTHFR and addressing that, which we can address it, it has a knock-on effect with all of these symptoms. COMT is an enzyme. COMT it has two primary roles. The one role is that it neutralizes estrogen, so it helps to clear estrogen, so you don't get those metabolites, those carcinogenic metabolites. But the other thing that COMT does is that it clears adrenaline and dopamine. So if, for example, you have a gene SNP in COMT, you might be somebody who doesn't sleep very well. Very often, I can chat to a patient and I can talk about what their life is like for them, and I can make an educated guess as to whether or not they've got this particular gene SNP. Often they're type A personality. They're usually very bright. They're good with the words. They're very dedicated. They are night birds because they don't sleep well. They can be a bit wired. They can have anxiety issues, because basically what happens is, because their body doesn't clear adrenaline, they have adrenaline in their body all the time. And when you've got adrenaline, it's your fight or flight hormone. So for them, they're on constantly high alert. So it affects stress, because if stress influences your risk of developing cancer, and you've got this COMT gene, you might have a double whammy, because you're not clearing the estrogen, and you've got stress, and with stress you've got belly fat, and of course belly fat increases your risk of cancer. So that's how the web feeds in, and you can see it's not linear at all. You have to address all the variables. But there's a tipping point. So you might find, let's say there are, I'm stuck here. Let's say there are 20 variables over which you'll get the condition. If you can address many of those variables, you never cross the threshold that takes you into a situation where you are manifesting a particular condition, despite the fact that you might have the gene SNP. So we want to address as many of those variables as possible, and they're all lifestyle things. You can address them all. So the clinical markers, these are all blood tests that one should go for at least every 2 years. Homocysteine is it's a bit of a surrogate one for methylation. It's a cheat test, because it does tell one, it's the best one we've got in South Africa. Actually, it's not ideal, but to see what the methylation process is like. If you want to see what's happening with inflammation, you could look at your high sensitive C-reactive protein. That's a blood test. Iron overload, so looking at ferritin, I'm going to talk about that now. We test to see whether you've got a gene SNP that allows the body to accumulate too much iron, and iron is extremely inflammatory. So especially for my male patients, men should have their iron levels, their ferritin levels checked, because lots of men accumulate iron. And it's one of the theories as to why women live longer than men. Also because women don't have dangerous activities, but it's because with regular periods, women clear their iron, whereas men don't. So men accumulate iron. And you'll see now, I'm going to show you a slide of all the impact, the conditions that can manifest if you've got high iron. Decreased vitamin D, you want to get your vitamin D levels checked. Your HbA1c, that looks at how your body's processing your sugars. Fasting insulin also looks at your sugars and whether or not you are insulin resistant. Thyroid, we've mentioned, because thyroid not only plays a role in Alzheimer's, but of course it also plays a role in your metabolic processes or your metabolic rate. So if you've got sluggish thyroid, you are more likely to gain weight and you're more likely to have obesity. And of course, your stress response, because a high stress response, we look at cortisol for that. Cortisol, I just want to say at this point, doing a morning cortisol blood test is a waste of money. Because when you're addressing stress and you're addressing cortisol, you really need to see what is the cortisol response throughout a 24-hour period, or really, it's pretty much the whole day. So to really see what's happening with cortisol, the best approach is to do 4 times in one day, salivary cortisol, and then you can map the cortisol response, and you can address the cortisol response when you know when the highs and when the lows are. All right, symptoms of iron overload. You can see there are many. Liver disease is one of them. Pancreatic disease is another one. Liver cancer, premature death, heart attack, testicular failure, all the cardiomyopathies. So heart attacks, hearing loss, diabetes. It affects your blood sugar. It affects your pituitary gland, osteoarthritis. I mean, I'm just skipping around here. You can read through them in detail. And it's such an easy one to test. You just test your ferritin. If your ferritin is very high, you go and give blood and it clears the ferritin. And it's obviously you've got to go through the process of having that needle, but it's easy to test. It's easy to fix. And the impact, the domino effect, the beneficial domino effect is huge if you have a look to see what impact iron can have in the body. So just to summarize, basically what this means is our genes are not our destiny. It's lovely to know what our genes are, because when you can see what your genes are, you have a direction. You know where to go, because we're all exposed to so much information that most of the time you just shrug your shoulders and you throw your hands up and you think, well, you know what, I'm exposed to so much information, I don't know which direction to take, so I'm going to rather do nothing. I'll just rather enjoy my life and hope that I'm going to live until I'm 95. So we can take steps in a particular direction. We can address the lifestyle factors, obesity, smoking, alcohol, a sedentary life, and toxin exposure, and we can tweak and pull them based on the genes. I do want to say at this point is that we can address the lifestyle factors without the genes. The genes are fantastic because they give direction, but it doesn't mean that if one is in a situation where you can't afford to do gene profiling, that you can't address the lifestyle factors, because of course you can, and you can make a great deal of difference in your life by addressing lifestyle factors. But I know, especially if you are somebody that you need evidence, okay, why must I address this particular one? If I'm going to give up smoking, or I'm going to give up drinking, I need a bit of motivation to do this, and gene profiling can just give you that extra bit of information. So thank you very much. I hope you've enjoyed this.